Hey guys, this is a review video of physiology on the thyroid hormone system. We get to learn all about the thyroid hormone today and how we get it made. We have iodine and iodide today, so nice and confusing. And then we have some follicles that we'll be talking about. So a little bit of everything. Starting with the overview, basically we have this hypothalamus pituitary axis. And so we have the hypothalamus is going to release thyroid releasing hormone, and that's going to act on the thyrotropes in the anterior pituitary and cause them to release thyroid stimulating hormone, which will go to the thyroid gland itself and have the thyroid glands from the follicular cells, these guys right here, they're going to produce thyroid hormone in the form of T3 and T4 to the blood. They'll be bound to carrier proteins. And overall, they're going to cause an increased metabolism, growth and development, and then an increase in the effects of epi and norepinephrine in the body. You also have a feedback mechanism on the anterior pituitary and the hypothalamus, and this is a negative feedback system. So us saying, hey, we got a bunch of T3, T4, why don't you please chill hypothalamus and please chill pituitary gland, stop releasing TRH, thyroid releasing hormone, and thyroid stimulating hormone. So not too bad. I've seen a bunch of this and maybe some histo, but what's going on with the microanatomy of the thyroid gland. So here you can see the thyroid glands on the trachea, and you have the right lobe, the left lobe, you have the isthmus, you have this little pyramidal part that comes up sometimes, not always there. But the unique characteristics of the thyroid are that you can palpate it. Cool. It's the only hormone, the thyroid hormone is the only hormone that needs an essential element from your diet, so iodine. And then T3 and T4 are stored in a proteinaceous, substance called colloid, which we find here. Okay, so this colloid substance we'll talk more about overall. We'll have some nice cleavage of T3 and T4 from its carrier in the colloid thyroglobulin. And what are the cells that are actually going ahead and doing this? Well, we have the follicular cells right here, and they're going to produce the thyroid hormone in the form of T3 and T4. They'll output it to the blood, and it'll get bound to some carrier proteins, of which we have 3 different ones that are helping out. And then we have calcitonin produced by our parafollicular cells adjacent to our follicles. All right, so calcitonin we are familiar with its effect on toning down calcium levels in the blood. All right, so then we have histological changes in response to being stimulated by thyroid releasing hormone. And so this is a typo. This should say thyroid stimulating hormone. That's my bad. Basically, what we have is our nice follicular cell, and when they have the thyroid stimulating hormone bind to its receptor, it is going to cause the follicular cells to elongate. These elongated follicular cells are going to be capable of producing more thyroid hormone. Okay, so low cuboidal, decreased secretion, tall columnar, increased secretion of thyroid hormone. Okay, so moving on, what are the actual structures of the thyroid hormones? We've talked about the fact that there's a T3, or there's, a T4 thyroxine, and this is the more stable version, and then you have triiodothyronine, T3, and you can see already that, if T4 is the more stable form, then the percentage of this hormone in the blood is much higher. But it does not bind to the T3 receptor very well, so its relative activity is very low. T3 is less stable, and so it is less prevalent. It will be T4 will be converted to T3 in the target cells, but T3 is doing most of the activity. And then, as we'll talk about, the sort of cleavage of T3 and T4 from the thyroglobulin that's makes up the colloid, we'll find out we have T4, T3 gets cleaved, but then sometimes you also get reverse T3, and this is the inactive form. It's very low in quantity, but it doesn't do anything. And basically, you can see the difference between these two is that the location of the iodide, IODIDE is slightly shifted, so reversed. But basically, just taking a look at these molecules, you can see we have 2 aromatic rings. Then we have, for T3, we have 3 iodine residues, and excuse me, this is incredibly tricky to keep track of. Iodide, IDE, is the ionic version with a negative charge, and you are inserting the form iodine, INE, onto these cells. So INE, dine, think of like to get ready for dinner, you are taking iodine, IODIDE, iodide, and then you're going to say, I am going to dine. So dine is second. Okay, so iodine, INE, is on these, and you can see that for T3, there's literally 3 of them, and then for T4, we have quite literally 4 iodine, right? After you get your food, you dine. But first you have to take in your food, which is iodide. Okay, so as we said, T4 gets converted into T3 in the target cells. This hormone is largely hydrophobic, which means that it has to, if we want to, keep it in the cell, it needs to be bound to something. And so when we're talking about the colloid, what we're really talking about, and I've mentioned this already, the thyroid globulin. So T3 and T4 are going to be stored as a protein conjugate with thyroid globulin. So you'll have this thyroid globulin backbone, and then you'll have, we'll see, DIT with another DIT, and we're going to get into this, but backbone of thyroid globulin with the T4s and T3s attached to it. Okay, and why does it need to do that? Because this hormone, with, you can see all these nice aromatic rings, is largely hydrophobic. Okay, because it's hydrophobic, when it does get into the blood, it's going to need bound to a carrier protein, and then since it can pass through cellular membranes, being hydrophobic, when it gets to its target, its receptor is going to be a nuclear receptor overall, so located inside of the cell, versus a membrane receptor for the proteinaceous hormones. This is a hydrophobic hormone, so nuclear receptor. All right, sorry, tiny bit of coffee. All right, initial steps in the production. So this is where it gets fun. We have, basically, we said thyroid globulin is going to be our backbone, and it's going to be something like this, and it's going to have some nice tyrosine residues, and then our cell is going to be like, hey, we need an essential element, and that's in the form of iodide. All right, so iodide is going to come in through this sodium iodide symporter, and it's going to basically take advantage of the sodium gradient to take in the iodide, and then that will take it into the follicular cell. And so the follicular cell is interesting because it is going to pump out the stuff that it just took in. And you're like, doesn't the follicular cell make T3 and T4? Yes, but it takes advantage of the lumen. And so basically what's going to happen is it's going to pump in iodide into here, and then that's going to be converted to I2, and then that's going to be added to thyroglobulin in here, and then you'll get this thyroglobulin with iodines on them, and then the thyroglobulin is going to go back into the follicular cell, and then at that point it's going to be cleaved into T3 and T4. Okay, so that's kind of the overview, but more specifically, we have iodine is being formed from iodine, but it has to go through 2 transporters first. The first is the sodium iodide, IDE transporter, and then the pedrin iodine transporter into the lumen of the follicle. Once it's in the lumen of the follicle, as a result of this pedrin transporter, the iodide, sorry, it's so hard to say this, iodide gets converted into iodine by thyroid peroxidase. And thyroid peroxidase has a couple of functions. The first is to take us from I- to I2, iodine, right? You set yourself up, you're making your little meal, and now it's time to dine. So dine is second. Okay, and then you have your thyroglobulin backbone is going to have all of these tyrosines on it, and then your iodine is going to get attached to that. And when the first attachment produces this molecule, we now call this tyrosine plus the iodine is now called an MIT. And what is that? Well, here's a little bit more detail. So this is connected to a thyroglobulin, and then you're going to have the sorry, so many typos. This is ine. Iodine is going to attach to the tyrosine on the thyroglobulin, and then this is attached to your thyroglobulin, and this portion on the thyroglobulin is called monoiodotyrosine. All right, so one iodine on a tyrosine, mono meaning one, so MIT. Okay, all right, and then the iodide that has been cleaved off of the iodine associates with the hydrogen atom. Okay, and next we have the production of the addition of a second iodine to our MIT on the thyroglobulin, and so that will produce something called a diiodotyrosine, so just two iodines. So what we're saying is we have this thyroglobulin, you're adding on sequential iodines to it, and building up the number of iodines to eventually we're going to get to a point where we're going to have 4 iodines on one molecule attached to thyroglobulin, and then you're like 4 iodines. That sounds a lot like thyroxine. What is thyroxine? Thyroxine is our guy right here, right? And so right here you would be attached to a nice thyroglobulin, right? And so you're gonna go chop, and now you have your thyroxine. Okay, but we're not there yet. So now we take our 2 monoiodotyrosines on the thyroglobulin, add another iodine, produce DIT. Now you have 2 DITs that are on our thyroglobulin, so 2 DITs on the thyroglobulin. They're going to go ahead and have a conjugation reaction, release some nice water molecule, and now you have a T4 on a thyroglobulin, okay? And so now you can cleave off a T4 to leave the cell. And you're like, okay, but what about cleaving off a T3? Is it possible to do that? Yeah, it should be. We didn't go over that in lecture, but you could imagine a monoiodotyrosine plus a diiodotyrosine being conjugated. Okay, so here's exactly what we just described, but in a different diagram. So first off, we have our sodium iodide, IDE transporter. It's taking an iodide into the lumen, and then it's saying, hey, we're just going to send it straight out. So send out iodide, IDE, into the lumen. Now that we're in the lumen, why don't we use thyroperoxidase to go ahead and convert that into iodine? And this transporter here is the Pedrin transporter. Okay, the follicular cell is also secreting the thyroid peroxidase, which is doing this work. Okay, now that we're in the lumen as iodine, we have our thyroglobulin that is in our lumen, and our thyroglobulin, which we usually see here, is so nice because it has all of these tyrosine residues on it, right? And look at those nice places where we could attack it with an iodine. All right, and so now we have iodination. Thyroid stimulating hormone also stimulates iodination of thyroglobulin. So you're going to have the formation of one of these guys into a monoiodinated tyrosine on this thyroglobulin backbone, the main chain of the thyroglobulin. Though technically this whole thing is thyroglobulin, right? But we're taking the sort of side chains of thyroglobulin and making them into MIT. All right, and then another iodine, again with our friend thyroperoxidase, you're going to get DIT. Okay, so then we've done this step, iodination of thyroglobulin. Now we're in conjugation of 2 diiodothyroglobulin. So we're going to take 2 DITs. So this is 2 DITs, and we're going to smush them together, and then we get a T4 molecule on our thyroglobulin. And now that we have a T4 on our thyroglobulin right here, we can go ahead and we're going to package this thyroglobulin into an endocytic vesicle back into the lumen of the follicular cell, and it's going to join other ones. And so basically, now that you're in the lumen of the you're in the lumen of the follicular cell, you are in this packaged lysosome, and you're going to have proteolysis of thyroglobulin. You're just going to chop these guys off, and so you might have some DITs, right? Maybe you have a thyroglobulin that has a bunch of T4s on it, maybe it has some T3s on it, and then it has DITs and MITs still on the thyroid globulin that haven't been made into, the side chains aren't entirely T4s and T3s yet. Okay, and so once you've chopped these guys up, you can send MIT and DIT back into the lumen with, so after you have some thyroid globulin synthase, you need to remake the backbone. But overall, not too bad. You've cleaved your T3 and T4, and they get exported. Okay, so again, the colloid containing the thyroid globulin that has T4 on it, T3 on it. Maybe it still has some DITs that have been made into that haven't been. So maybe these two guys haven't been combined into a T4 yet, and then maybe you still have some MITs on it. This is going to be cleaved. All right, and so when it's cleaved in the lysosome, then you have your MIT and DIT. Oh, yikes, this is a typo. I'm so sorry. MIT and DIT will get deionated, and then they can go back into the lumen of the overall follicle, and then T3 and T4 can leave the cell overall. Okay, so not too bad. When the T4 is secreted, you have basically, it will be, as we said, converted into T3 in the target cell. About 35% becomes T3, 10% of T4 gets destroyed, and then we were told 45% becomes reverse T3, but that's inactive and then basically broken down. Okay, so sources of circulating T3, most of it's coming from T4, some of it just comes directly as T3, but it's because it's hydrophobic, it has to be bound to carrier proteins, and so 99% of them are in fact bound to some sort of plasma protein. We have three different ones. We have thyroxine, which T4 binding globulin, and that holds 45 to 60% of T3, but 75% of T4. Then you have thyroxine binding prealbumin, and that does a little bit less, 15% to 35% of T4, but it does not do T3. So prealbumin does not do T3. This is also called transthyretin. Okay, transthyretin is also called thyroxine binding prealbumin. Then you have albumin itself, which binds T3 and T4, but thyroxine binding globulin, thyroxine binding prealbumin, which doesn't do T3, and then albumin itself, which does T3 and T4, but in lesser amounts. But it's mainly thyroxine binding globulin. All right, so then we have the hypothalamic pituitary axis. And we've already talked about this, but our TRH is released by the arcuate nucleus. That's going to come down and go to the anterior pituitary, and our anterior pituitary is going to, you remember B flat from our histo lectures? Well, what is flat? Flat is follicle stimulating or thyroid stimulating hormone, right? So this is coming from the basophils of the anterior pituitary, and but before we go there far, we have arcuate nucleus is releasing TRH into these nice portal veins. That's going to go to G-coupled protein receptors in our thyrotropes in the anterior pituitary. That's going to activate this DAG, PCK, PLC, and IP3 pathway, increasing protein phosphorylation, releasing calcium stores. The calcium store is going to trigger the synthesis of thyroid-stimulating hormone, also called thyrotropin. Thyroid stimulating, I really hope they don't ask us about that. That's so uncool. Thyroid stimulating hormone increases, we know, T3 and T4 from the follicular cells, and then you have negative feedback on both the thyrotropes in the anterior pituitary and on the nuclei in the arcuate nucleus. Okay, and so T3 and T4 are doing negative feedback, and then you also have somatostatin and dopamine can inhibit the thyrotropes as well. So dopamine, thyrotropes, I don't think somatostatin is labeled here, but here's just a nice tyrosine stimulating hormone from anterior pituitary binding to G-coupled protein receptor on the follicular cell, stimulating uptake of iodide, IDE, and then export of tyrosine peroxidase and synthesis of thyroglobulin. All right, so then cellular action, what does this actually do? It enters the target cell. T4 is cleaved to T3 by 5,3-mono-deiodinase enzyme. All right, well, that makes sense because you need to get rid of an iodine. And so you have that occurring here. So T3 coming in, T4 coming in, T4 being converted into T3 by monodeiionidase. T3 is going to go to its nuclear binding receptor, and it will form a complex with another receptor called the retinoid X receptor, RXR, and they're going to bind to the thyroid response element in the promoter region and cause an increase in gene transcription, which is what this is all saying. Okay, physiological effects of thyroid hormone. So basically just think increased metabolism. And then you're like, how does it do that? Increased metabolism in all shapes and forms, but with a net result in energy like output. All right, so increased metabolic activity, increased glycogenesis and glycogenolysis, no change in glycogen stores. So increase the production of glycogen, increase the breakdown in glycogen overall, increase in gluconeogenesis, so pump out some glucose, decrease protein production, increase lipogenesis and lipogenolysis, and so you have a slight favoring of lipolysis and therefore reduction in fat stores with T3 activity, increased rate of cholesterol synthesis and degradation with the net result of a decrease in plasma cholesterol as you have LDLs being taken up into the liver and the net production of the good cholesterol HDL. T3 is required for normal growth and development. Hypothyroidism causes intellectual disability, and congenital is from birth. So from birth, hypothyroidism, if you have it from birth, intellectual disability. During development, you need to have T3 present in the first 3 months, or you're going to have intellectual disability that's permanent. And then T3 in excess during development will cause premature closure of the epiphyseal plates on your long bones, and that will mean stunted growth. Okay, T3 on the nervous system. If you have a deficiency in adults, you're going to be sleepy, listless, weak. It's a decrease in nervous system function. If you have an excess, you have hyper-excitable nervous system. You're increasing T3. One of the things we said at the very beginning was it increases the epinephrine receptors, so increases sensitivity to beta-adrenergic receptors. All right, pathologies, simple goiter, just an enlarged thyroid gland, potentially normal hormone secretion, can be caused by iodine deficiency. Hyperthyroidism is basically, we talked about Graves' disease, where you have autoimmune stimulation of the thyroid stimulating hormones. So you have too much T3 and T4, and typically that's a result of a thyroid tumor or a pituitary tumor on your anterior pituitary. Hypothyroidism, we learned about Hashimoto's, and that is autoimmune destruction of the thyroid glands, not good. But for children, you have intellectual disability, decreased growth rates. Adults, not having enough T3, T4 means decreased metabolism, sluggishness, cold sensitivity, increased sleep, thick and dry skin. What would cause hypothyroidism? Poorly developed thyroid glands, impaired sensitivity to thyroid hormone, due to a rare genetic mutation, increased production or reduced production of thyroid hormones caused by a defect in the production system, and then, as we said, Hashimoto's. All right, so that's it. Unfortunately, we have exam tomorrow, so good luck, guys. The L3 or 35, 36 parathyroid lectures, just don't have enough time to make a review video, so I'd suggest using the MLB review notes, but good job, guys, and you can do it.